May peace be on all of you. Myself, Dr. Majid Khan, Associate Professor, Loknitya Dr. J.D. Pawar College of Pharmacy, Kalwan, Nasik. I welcome all of you in this new chapter, Antidiabetic. In this video, that is in lecture 1, we will cover insulin and its preparation. So the learning outcome of this topic is to be able to learn about the insulin structure, their biosynthesis, their structure activity relationship, and various form and preparation of insulin available in market. So what do you mean by diabetes? The word diabetes is a Greek for siphon applied to this condition because one of its symptoms is excessive thirst. The other symptom of diabetes mellitus is weight loss, excessive hunger, and presence of glucose in urine. In 1869, Paul Langerhans observed that pancreas consists of two types of cell. The acinar cell secrete digestive enzyme and cell cluster like islate which had unknown function. In 1889, Banting shows that serum glucose level were associated with pancreas. This shows that pancreatic extract could reduce serum glucose level. In 1922, the pancreatic extract was administered to 14-year-old patient exhibiting serum glucose level of 500 milligram per deciliter who survived 15 years on insulin obtained from animal source. This success led many to conclude that diabetes is a disease of elevated blood sugar easily corrected by exogenous insulin. There are two types of diabetes, that is type 1 called as insulin dependent diabetes mellitus. Is the interleukin, which is a protein which trigger all immune responses. This substance is produced by macrophage as a result of antigen processing. It mobilizes B and T cell and stimulates T helper cell to produce interleukin 2, important for immune response. It is seen that pancreatic islate of animal with spontaneous diabetes have shown to contain inflammatory cell incorporating interleukin 1 and incubation of islate with interleukin 1 selectively destroy beta cell. This fact indicates that type 1 diabetes probably result from the autoimmune process involving interleukin 1, but the reason why production of interleukin 1 is increases is not known. In some cases, viruses are involved in production of type 1 diabetes. One of the most convincing involved previously healthy young boy who suddenly developed very severe diabetes and died after 7 days. Then second type, that is type 2 diabetes, that is non-insulin dependent diabetes. It occurs more commonly in obese individual. Overeating coupled with underactivity leading to obesity. It is associated with development of non-insulin diabetes mellitus. This is due to decrease in insulin receptor on insulin response cell. The patient may have either normal or elevated insulin level. It is suggested that overeating causes increase in insulin production but decrease synthesis of insulin receptor. So this type 2 diabetes is more common. Now what do you mean anti-diabetic agent? So these are the agents which are used in diabetes. Then insulin, that is biosynthesis and structure of insulin. Insulin is synthesized by isolate of beta cell from a single chain 86 amino acid polypeptide precursor called as proinsulin. Proinsulin itself is synthesized in polyribosome of rough endoplasmic reticulum of beta cell from an even larger polypeptide precursor preproinsulin containing 110 amino acid. The proinsulin is enzymatically converted into insulin within the pancreatic cell. So first, pre-proinsulin, which contain 110 amino acid, converted into pre-insulin, which contain 86 amino acid. Then this proinsulin, then converted into insulin. So first start from the pre-proinsulin, which contain 110 amino acid. Pre-proinsulin, which contain 110 amino acid, converted into proinsulin, which having 86 amino acid, and then this proinsulin is converted into insulin. So here there is a structure of proinsulin which contain 86 amino acid. So here the proteolytic conversion of proinsulin to insulin is accomplished by a removal of arginine residue at position 31, 32 and 65, and there is a lysine at 64 by endopeptidase enzyme. So here there is a structure of proinsulin which contain 86 amino acid. So here there is a chain. First this black, it is called as B chain. Then it is a gray color, it is called C chain. Blue color, it is called A chain. So there are total 86 amino acid, and it is the structure of proinsulin. So here there are two sites where the cleavage is taken place, at this site, and second one is this site by the endopeptidase enzyme. So after this breakage we get four products. First one is, that is, one molecule of insulin which contain A and B chain, one molecule of C peptide, and four basic amino acid. That basic amino acid is arginine 31, arginine 32, arginine 65, and lysine 64. So after cleavage of this proinsulin which contain 86 amino acid, so here there is a breakage. First breakage is here, second breakage is here. So after this breakage we get this four product. First product is insulin, one molecule of insulin which contain A and B chain. This one, that is blue, and this one is black chain. It is called as insulin. Combination of A and B chain only, it is called as insulin. Then second product is this C chain, that is called as C peptide, and there is a 4 amino acid. In that there is a 31, 32, 65 arginine and lysine at 64, like this. So after breaking we get one molecule of insulin, that is A and B chain, then this C peptide separated, then there are two bases, and here there are two bases. Total 4 bases we get. So the resulting insulin molecule consists of chain A and chain B. Okay, so in case of insulin we get one molecule of insulin with A chain and B chain. A chain is from 66 to 86. So here from A chain, blue color, 66 to 86 there are 21 amino acid. Then B chain, black one, that is from 1 to 30 there are 30 amino acid sequence. So insulin structure consists of A and B chain. In that A chain consists of 21 amino acid, B chain consists of 30 amino acid. Apart from that, insulin contains disulfide bond. So it is a disulfide bond. There are 3 disulfide bond. first disulfide bond is from the amino acid 7 to 72, this 7 to 72. Then second one is 19 to 85, 19 to 85. It is a connection between this disulfide bond from A and B chain. And third disulfide bond is within the A chain, this one, that is from 71 to 76. So it is the connection between the A and B chain through the disulfide bond. Now the receptor binding site in A chain, so in case of insulin there are receptor binding site. So the receptor binding site region consists of glycine A chain. In A chain there is a glycine 1, glutamine 4, glutamic acid 5, tyrosine 19 and aspargine is 21. So these are the main receptor binding site on A chain. Similarly, in case of B chain, valine 12, tyrosine 16, phenylalanine 24, tyrosine 26 on B chain. So these are the receptor binding site on A chain and B chain of insulin. So it is the biosynthesis of insulin. So in short, proinsulin, which contain 86 amino acid, is cleaved by the endopeptidase enzyme, and after cleavage it gives 4 products. First product is insulin, which contain A and B chain. Second product is C chain, that is called as C peptide, and there are 4 amino acids separated, that is in that 4 amino acid 3 is arginine, that is 31, 32 and 65 number, and lysine at 64. So it is the biosynthesis of insulin. The amino acid sequence of insulin from various animal species have been examined, and detail of this is as follows. So this table shows the insulin from the various source. For example, here first column species is given: human source, cow source, pig, horse, rabbit, dog, chicken and duck. Here there is a difference of insulin from various sources on the A8 position. A8 position means on chain A, 8th amino acid. Then A9 means 9th amino acid in A chain. Similarly A10, then B1 means first amino acid of B chain. Then B2, B27 and B30. So here if we see the human insulin, the insulin which is obtained from the human being, the sequence on A8 position there is a threonine, on A9 position there is a serine, then A10 position there is a isoleucine, on B1 phenylalanine, B2 valine, B27 threonine, B30 threonine. In case of insulin obtained from the cow, so here there is a difference. In case of A8 position there is a alanine instead of this threonine. Similarly here there is a valine instead of isoleucine. Similarly here last alanine instead of threonine. Similarly in case of pig, if you see the in case of pig only one difference is there, that is on B30 there is a alanine instead of threonine. Similarly in case of horse there is a 2 differences, glycine and alanine. Then in case of chicken there are lots of differences amino acid sequence. So these are the all difference in the sequence of various species insulin. It is found that hormonal sequence for porcine insulin means pig insulin, is called as porcine insulin also. So it is found that hormonal sequence for porcine insulin is closest to that of human, difference only by substitution of alanine residue at C terminus of B chain instead of threonine. So in all these species insulin, the pig insulin, also called as porcine insulin, it is closest resemblance to human insulin. Only one difference is, that is alanine instead of threonine at the B30 position. So porcine insulin, or called as pig insulin, having the maximum structural similarity with human insulin. Therefore, porcine insulin can be converted into human insulin. It should be kept in mind that more dissimilar the sequence, greater is the potential for antigenicity. This bovine insulin is more antigenic than porcine insulin in human. So when there is a more difference in structure sequence, there is a more chances of side effect, that is called as antigenicity. So in case of pig or porcine insulin, there is a less antigenicity because it having the amino acid sequence approximately same as human insulin. The A and B chain were combined to form insulin in 60 to 80% yield. Nowadays, recombinant DA technology had been applied successfully in production of human insulin on commercial scale. Human insulin is produced in genetically engineered E. coli. There are two available methods of applying recombinant DNA technology in production of human insulin. The first method involves insertion of gene for production of either A or B chain of insulin molecule into the special E. coli strain and subsequent combining two chain chemically to produce insulin. That is structure and chemically identical with human insulin. In second method involve insertion of gene for entire pro-insulin molecule into special E. coli that are then grown via the fermentation, then completing C peptide is then enzymatically cleaved from the pro-insulin to produce human insulin. So recombinant technology is utilized nowadays to produce the insulin. So there are two methods. The first method, so here there is a first method, A and B chain synthesized separately and then chemically they are joined. And second method, that is gene for pro-insulin is directly inserted into E. coli and then there is a C peptide is separated chemically. So these are the method for insulin production. Now SAR of insulin structure activity relationship of insulin. So in that first point, amino acid, AA stand for amino acid. So amino acid from chain A cannot be removed without loss of activity. So as we know, insulin consists of A and B chain. So in case of E chain, there is a 21 amino acid. If we remove any one amino acid from A chain, the activity is lost. Second, B chain up to first 6 and last 3 amino acid can be removed without decreasing activity. So as we know, B chain consists of 30 amino acid. So first 6 and last 3, if we remove this amino acid, the activity retained. Third point, two insulin analog are prepared by replacing A glycine, chain A glycine with L and D alanine. So as we know, A chain, in case of A chain, the first amino acid is glycine normally. In case of human insulin, A chain amino acid, that is first amino acid in A chain is glycine amino acid. So if we replace this glycine with L and D alanine, capital L and D alanine, so it is the isomer. L, capital L means when amino acid is left side, D means it is right side. So glycine is replaced with L and D alanine and insulin is synthesized by this way. So it is observed that the L analog is 9.4% and D analog 95% as potent as insulin in glucose oxidation. The binding affinity to isolated fat cell is 10% for L analog and 100% for D analog. So two form of insulin is prepared by replacing glycine of A chain with L and D alanine. So it is found that there is a less activity as compared to normal insulin. L analog only show 9.4% activity while D analog show 95% activity of normal insulin. So the substitution of alpha carbon of A chain of human insulin with CH3 group is in particular configuration interfere with binding with receptor, hence resulting an analog is much less active. Methyl substitution in opposite configuration affect neither binding nor bioavailability. So here glycine, which is present in normal insulin, and alanine, here there is a CH3 is present. So only difference of this CH3. So the configuration of this CH3 is very important. So in case of D-alanine, configuration of this CH3 changes. There is a configuration is difference between D and L-alanine. So here D-alanine is more potent than the L-alanine insulin. Next point. Removal of N-terminal phenylalanine from B chain produce compound with activity. So if we remove phenylalanine from the B chain, then compound show the activity. Next point, the single residue deletion at position B24 through B26 provide analog about twice as potent as insulin. So in case of B chain from 24 to 26 amino acid, if we delete or if we remove the amino acid, then it shows the twice activity as compared to insulin. 6th point, when B26 tyrosine was replaced with phenylalanine and B24 phenylalanine replaced with D-tyrosine, provide a compound twice potent. So here there is an exchange of amino acid. Normally B24 is a phenylalanine and B26 is tyrosine. So if we exchange this amino acid, the activity is twice increases. While B26 tyrosine was replaced with phenylalanine and B25 phenylalanine replaced with D-tyrosine, compound is inactive. Then 7th point, several authors have reported that active compound can be obtained by removing B27 to B30 tetrapeptide. It means that if we remove the last 4 amino acids from the B chain, then the compound is active. So these are the about the SAR of amino acid. Now next point, which is important one, that is form and insulin preparation available in market. So there is a difference. Biosynthesis means formation of insulin within the body by using proinsulin. Here preparation of insulin means the forms, various forms of insulin available in the market. So when question is asked about the preparation of insulin, so you have to write that is various form which is available in market of insulin. And if question is asked about the biosynthesis, then you have to write the biosynthesis process, that is from proinsulin to insulin. So now here is the forms of insulin. So amorphous insulin was first form made available for clinical use. Further purification afforded crystal insulin, which now commonly called regular insulin. Insulin injection USP is made from zinc insulin crystal. Sometimes a regular insulin solution have been prepared at pH 2.8 to 3.5. If pH increase above the acidic range, particle would be formed. Neutral insulin solution have greater stability than acidic solution. Neutral solution maintain nearly full potency when stored up to 18 months at the temperature 5 to 25 degree. The major disadvantage of regular insulin is short duration of action, that is 5 to 7 hours, which necessitate its administration several times daily. So the first form of insulin, that is amorphous form. Then after development in purification technique, another form is come, that is crystalline insulin, also called as regular insulin. This regular insulin is prepared at the pH 2.8 to 3.5, that is acidic pH. But if the pH increases above the acidic range, then the impurity will appear, that is called as particle would be formed. Then second, neutral insulin is prepared, it having the more stability than the acidic solution, and it maintains the full potency when stored up to the 18 months at 5 to 25 degrees Celsius. But the major disadvantage of this regular insulin is short duration of action. It only gives the action up to the 5 to 7 hours. Therefore, which necessitate its administration several times daily. Because it having the short duration of action, the patient may take the dose number of times during the day. Therefore, in order to overcome this short duration of action, many attempts have been made by prolonged duration of action of insulin. For example, development of insulin form less water soluble than highly soluble. Protamine insulin preparation proved to be less water soluble and less radially absorbed from body tissue. So protamine is a new insulin which is less water soluble and having the longer duration of action as compared to previous insulin. Then protamine zinc insulin suspension, that is called as PZI, protamine zinc insulin suspension, where even longer acting, that is 36 hours as compared to the protamine insulin. The PZI is prepared by mixing insulin protamine and zinc chloride with buffer solution. After that, isophane insulin suspension. Next form, isophane insulin suspension, also called as NPH. So isophane means, iso means equal, phane means appearance, and NPH stands for N for neutral, P for protamine which is amino acid, and H for hagedron which is a scientist name. So isophane insulin, or also called as NPH, has quicker onset of action and short duration of action than PZI. NPH is given in single morning dose and normally exhibit greater activity during the day than at night. NPH and regular insulin can be combined for many patients with diabetes. Now, the major concern or disadvantage of PZI and NPH insulin is potential antigenicity of protamine, which is an amino acid obtained from fish. This leads to development of lente insulin. So this leads to the new type of insulin, that is lente insulin. This insulin, by varying the amount of excess zinc by using acetate buffer and by adjusting pH to, so there are two types of lente insulin were prepared. At higher concentration of zinc, microcrystalline form precipitate and called as ultra lente. It is relatively insoluble and has slower onset of action and longer duration of action than PZI. At low zinc concentration, amorphous form precipitate, and it is called as semi-lente form. It is more water soluble, quicker onset of action, shorter duration of action than the regular insulin. And the third type of insulin suspension, that is lente insulin, which contains 70:30 mixture of ultra plus semi-lente insulin. This lente insulin has a rapid onset of action and intermediate duration of action. Lente insulin are chemically incompatible with PZI and NPH insulin because different buffer solution used in the preparation of these insulin, that is acetate buffer in lente and phosphate buffer in PZI and NPH. Additionally, regular insulin will be remain fast acting when combined with NPH, but not when added to lente. The rapid action of regular insulin is neutralized by excess zinc present in lente insulin. So these are the various forms available in market. So again I repeat, first form, that is amorphous form, older form amorphous after purification technique, crystalline form discover or synthesize. This regular insulin, that is called as crystalline form, is prepared at the pH 2.8 to 3.5, but if the pH increases above the acidic range, then the impurity will come. The next form is prepared, that is called as neutral insulin, which having more stability as compared to acidic form. But the disadvantage of this regular insulin is short duration of action, that is 5 to 7 hours, which necessitate its administration several times daily. So to overcome this short duration of action, other form of insulin is developed. In that first is protamine insulin. Protamine is an amino acid. It is a less water soluble and less radially absorbed from the body tissue. Then after that, protamine zinc insulin suspension is prepared, called as PZI. So it contains zinc. So it is prepared by mixing of protamine insulin plus zinc, called as PZI. It having longer acting, 36 hours, than the protamine insulin. Then after that, isophane or NPH insulin come. So isophane means, iso means equal, phane means appearance. NPH, it is a short form of N for neutral, P for protamine, H for hexadron, which is a scientist name. So this NPH and isophane insulin is a quicker onset of action, short duration of action than PZI. It is given in single morning doses and normally exhibit greater activity during the day than the night. NPH and regular insulin can be combined for many patients with diabetes. So it is also given in combination. The major disadvantage of PZI and NPH is antigenicity, and it is due to the protamine, which is an amino acid obtained from the fish. So the major disadvantage of this above two, PZI and NPH, is antigenicity due to this protamine. So to overcome this, new form, that is called as lente insulin, is prepared. So there are two types of lente insulin. First one is ultra lente insulin, which contain high concentration of zinc, insoluble and slower onset of action and longer duration of action. Second one is semi lente, which contain lower concentration of zinc, which is amorphous form, more water soluble, quicker onset of action, shorter duration of action than the regular insulin. Third one is lente insulin, which is a mixture of above two, that is ultra plus semi lente at the ratio 70-30, and it having the rapid onset of action. Lente insulin is chemically incompatible with PZI and NPH. So this type of insulin, called as lente insulin, it is incompatible with previous two, PZI and NPH, because of difference in the buffer used in the preparation. So these are the various form which is available of a insulin form. Now in market, these all form are divided into four category. That is rapid acting, short acting, intermediate acting, and long acting. So see one by one. So in case of rapid acting, first one is insulin aspart, called as Novolog. In this Novolog, which is given in the bracket, it is a trade name available in market. So when you go to medical, then if you ask about the Novolog, so it give this type of insulin, insulin aspart. So this in bracket, it is a trade name. So insulin aspart, it is a long rapid acting insulin, same with the regular human insulin, except that proline was replaced by aspartic acid in position B28. So in case of insulin aspart, the aspartic acid at the B28 is there instead of proline. So proline B28 is replaced by the aspartic acid in this type of insulin. Onset is 15 minutes, duration is 2 to 4 hours. Second example of rapid acting insulin is insulin lispro, that is called as Humalog. It is a trade name Humalog. So here also B28 lysine and B29 proline of human insulin analog, where is the usual B28 proline and B29 lysine is reversed. So here there is an exchange of amino acid sequence at the position B28 and B29. Onset of action 15 minutes, duration 2 to 4 hours. Rapid acting, another example is insulin glulisine. It is another insulin analog with asparagine at position B3 replaced by the lysine. So here again asparagine replaced by the lysine at the B3 position and lysine at B29 replaced by the glutamic acid. Then duration of action is same as previous insulin. Then there is a next class, short acting insulin. So here example, a regular insulin, trade name Humulin or Novolin. The only insulin that approved to be administered via IV route come from pork or porcine, beef that is called as bovine, or human pancreas, or genetically engineered using recombinant DNA technology from certain microorganism that is E. coli. Onset of action 30 to 60 minutes, duration 6 to 8 hours. Then intermediate acting insulin, that is isophane insulin. So it is, as we learn, that is isophane or NPH insulin or NPH insulin. So it is a sterile suspension of zinc insulin crystal and protamine sulfate, a protein from the sperm of fish. Duration, onset is 1 to 2 hours, duration 10 to 16 hours. Next example, insulin zinc suspension called as lente insulin, as we discussed. It is a sterile suspension of insulin modified by the addition of zinc chloride. It has advantage of fewer allergic reaction than NPH. Onset 1 to 2 hours, duration 10 to 16 hours. Then long acting extended insulin zinc suspension, that is ultra lente insulin. It is a sterile suspension of insulin modified by the addition of zinc chloride. Onset of action 4 to 8 hours, duration 36 hours. Insulin detamir is another long acting insulin analog that may have some benefit over the isophane insulin. It is a neutral soluble human insulin analog in which the terminal amino acid at B30 has been replaced by myristic acid. It is a 14 carbon fatty acid chain. Duration up to 24 hours. Then insulin combination. Sometimes combination may be available in the market. So here there is a pre-mixed, means पहले से जो mix किया है, that is pre-mixed. So it is a combination of Novolin, that is 70-30 ratio, contains 70% NPH and 30% regular insulin. So this Novolin is an example of pre-mixed insulin mixture. So it contains two form, that is NPH 70%, regular insulin 30%. Similarly, another example of pre-mixed is Humulin, 50-50 NPH and 50 is regular insulin. So it is a summary of various form of insulin. It is a trade name, it is a generic name Lispro, and it is a trade name depending upon the company. Here the difference, if in any difference of sequence of insulin as compared to human insulin. Similarly here, for example, first one is Lispro, it is a generic name. Humalog, it is a trade name. A chain, there is no changes, but in case of B chain, B28 proline is replaced by lysine. Similarly, B29 lysine is replaced by proline or protamine. Then aspart, that is Novolog, it is a trade name. Then here there is no changes in A chain, but here there is a changes. B28 protamine replaced by asparagine, then B3 valine replaced by the lysine, and B29 lysine replaced by the glutamine. Then detamir, that is levimir, is a trade name. Here there is a changes in A chain also. A21 asparagine replaced by glycine. Sorry, in case of levimir there is no change, but here there is a change in B chain. Remove B30 threonine and addition of a C14 fatty acid to B29 lysine. So it is a in short summary of some types of insulin analog. So these are the various form and preparation available in market of insulin. So it is about the biosynthesis. There are various form available in the market. So if you like the video, then subscribe the channel. Thank you very much.